Well, I thank the organizers, but it's probably not the right thing to do. Anyway, thanks for everybody for staying around so long. You've heard some great talks today, and I'll try to be brief so that we can get to our final discussion. I work on congenital disorders of glycosylation. Glycosylation is this physiological process of putting sugar chains onto proteins. And I'm calling this Stories from Our Village. And as you see, there's some good news, and there's some news that we kind of wish hadn't been quite the way it was. But anyway, we're getting some lessons that we hadn't expected. And a while back, the number of glycosylation disorders was just very few, a couple a decade ago, and now there are over 40. And so they're really beginning to expand a lot. And we managed to get this student, new postdoc in the lab, to come in and point out the new disorders. And this is sort of a perspective of glycosylation that occurs in the endoplasmic reticulum. And every place you see a red X there, that is a location where a mutation in the N-glycosylation pathway has been found. And the red pen there marks the most recently discovered types of disorders. The thing that is perhaps interesting here is that we're not going to talk about all these. This is in fact only half of them. We're in fact going to focus down just in this little group down here. These are ones that are called CDG, Congenital Disorder of Glycosylation, 1A and 1B. And a term that you're going to hear, fairly common, probably over and over again through the talk, is right here.
Mannose 6-phosphate.
What was that? Say that again?
Mannose 6-phosphate.
So, you heard the man. We're going to hear Mannose 6-phosphate a lot. And so this is the metabolic pathway for mannose, and mannose is a major component of glycoproteins. And what you see is that mannose can come in here into the cell and be converted by hexokinase to mannose 6-phosphate. And it can also be made from glucose via fructose 6-phosphate and phosphomannose isomerase. This makes mannose 6-phosphate, which is then converted to mannose 1-phosphate by phosphomannomutase 2, and then there's a whole bunch of other reactions that go on, and that's what makes the glycoproteins. So we're not going to worry about all those other ones here at the moment, just these couple of enzymes. Well, it turns out that the very great majority of mannose in glycans actually is derived more from fructose 6-phosphate, in other words, glucose, than it is from mannose. And if you look, you can see that the ratio of these two enzymes, PMI or MPI, either one, to PMM, dictates what will happen to mannose 6-phosphate. And it makes sense because those are the two enzymes that are essentially competing for the same substrate. So you raise one, you lower the other, and you can affect the sort of the metabolic flux. So here is what I'm calling the good. We were able to find a patient who was defective in this enzyme, PMI, CDG-1B, and we were able to give this little boy mannose and rather quickly reverse all of his pathologies, and that was not hepatic fibrosis, but protein-losing enteropathy, coagulopathy, hypoglycemia, all these things reversed when he was given mannose. Well, why did we give him mannose? We had a paper out earlier that suggested if you had cells of some glycosylation disorder, and at the time we didn't know the defect, if you had cells, glycosylation was bad, you simply added mannose to the culture medium, the glycosylation in those cells improved. And so we thought, well, wouldn't that be great if you could do this with patients? A number of people in the lab, some are still here in fact, went down the hall with FDA approval, and we drank mannose to see if we could absorb it and whether it could be incorporated into proteins and into the blood and all, and it seemed to work. So we actually had a call from this little boy's doctor, and he said, I have a patient who's probably going to die. He's lost an enormous amount of blood. Do you have any idea how much Mannose to give, and would it have any effect? I said, well, I don't know if it's going to work, but given this much, this often he'll achieve this high of a level, it should clear out at this rate. And as it turned out, it actually did. But we didn't know what the disease was. Well, as it turns out, he was deficient in this enzyme right here, phosphomannose isomerase. He was not a null, he was just deficient. So the therapy then really consisted of giving Mannose, which was just able to bypass that particular block. And since 95% is normally catabolized and only something like 2 to 4% of the Mannose coming from outside is used for glycosylation, we were able, just by upping the exogenous concentration in the plasma by about 3-fold or so, to drive enough Mannose into glycosylation to take care of this. I mean, that was really quite amazing to us, and we celebrated, and that clearly was the good. However, CDG-1B is not the major type of glycosylation disorder that most patients have. In fact, what they have is CDG-1B, and that's this enzyme. That's the one that goes after the creation of mannose 6-phosphate and conversion into mannose 1-phosphate. Now, Mannose is not effective for these patients, and the reason is because, of course, mannose 6-phosphate is before the block, right? And so now you can't get past the block by giving Mannose. But we had some other data from a long, long time ago that suggested that maybe there would be a concept that we could use, and that is if you took cells from a CDG-1A patient and looked at the amount of glycosylation, and that's what's plotted here. This is a control at 100%, and these are 2 different CDG-1A patients. You can see that the glycosylation is considerably lower. If we give Mannose, fairly high concentrations of Mannose, that you can actually bring the level of glycosylation up to normal. That's in a patient who has a partial deficiency in phosphomonomutase. So we got the idea that maybe what you could do is, since many patients will have between 2 to 20% residual phosphomonomutase activity, in other words, that enzyme right there, and even those with 25% activity are patients. They're affected. And there are parents who have 50% activity, and they're fine. Where is that break point? If you went from 25 to 37.5% activity, would you be able to get something? Could we just in some way activate or promote that enzyme in a way that would give us some more benefit? Well, so normally what happens is exogenous mannose comes in, and most of it's degraded. Degraded meaning that it goes into glycolysis. But let's suppose we could shrink down phosphomannose isomerase. Now what would happen is that mannose 6-phosphate is available to go into the glycosylation pathway, and it might improve the glycosylation. So this was a direction we went, and there's a year and a half that I'll summarize in a couple seconds, and basically we did a high throughput screen at the Conrad Previs Center for Chemical Genomics, in other words, a building just next door, and we were able to come up with a few compounds that did the sort of thing we wanted to. And I won't go through all of the details on that, but we were able to, at least in principle, show that this could work. And so if you look at one of our CDG-1A patients, and we didn't have all the patients like Brandon just talked about, we only had a limited number, we picked this one, and as you can see, when we add this compound we called HT10, high throughput screening compound 10, but there were 200,000 that went through the screening, so it's not quite that simple. You can see that when we add this compound and we do a measure of glycosylation, as we increase the mannose, in fact, more of the mannose is able to go into glycoproteins here at a lower mannose concentration, and as for these 2 concentrations, and as you go up, you finally saturate the system. This says that you're more efficiently using the mannose that's in the medium, and you have a chance of improving glycosylation. So we were quite happy to see that, and in fact, we looked at a number of other different CDG lines, and we found that these two were especially good, but as you heard in Brandon's talk, not every patient mutation would respond equally to therapy. So these two responded quite well. A number of these other ones haven't, and we still have more to check. But at least the proof of principle is there, and we think that we may have a ways to go, but we'll be able to improve on this since we're now doing the crystal structure of phosphomannose isomerase with and without the compounds. So this ought to work on KM mutants of PMM2 and mutations affecting the stability and folding, and maybe with some of Bill Balch's help, we'll find some other approaches to this. So we decided then that we're going to now go back to CDG-1b, and because we're basic scientists and we wanted to understand the process of glycosylation and how this was working, and since there were a few patients, and it's ethically not all right to work on patients, of course, we would make a mouse model. And so what we did was to completely knock out phosphomannose isomerase. And this was a project that Charlie DeRossi did in the lab, and the problem, here we go, the problem was when we did that, it was lethal. So we completely did away with all the activity, and that's the only gene that can carry out this reaction. So you say, well, you forgot to add mannose. If you just would feed mom some mannose, then the embryos wouldn't die. Well, that's what we thought too, and when we did that, of course, what happened is that the embryos died earlier. And then we were puzzled. What was really going on? Well, it turns out that when you do that, you accumulate mannose 6-phosphate because it has no other place to go. It comes into the cell and it's sitting there. And what that does then is to deplete the ATP within the embryo, and the embryo dies. And so this was not really a very useful model. So what we had to do then was to find a patient mutation, knock that into the mouse, and then try to use that as a model system. So this is what we did here, and now we have the new good. Here's the normal mouse, and here is a heterozygote. Of course, the total null over here with 0 activity is lethal, but we could make this knock-in mutation. If we had 2 of those mutations, we ought to have 16% activity, and if we cross that with the knockout model, then we would have 8%. So this was good because it was in the range that we would expect to have for patients. About 5 to 15% activity is typically what you see. And so when we did that, this Bobby Ning and Vanana Sharma worked a lot on this and showed that, in fact, it gave you a lot of what we predicted. So this was the predicted amount here, about 15 and 8%, and you can see that in a number of different cells and organs, we got that. But then there were some tissues that were hit much more drastically. We said, oh, that's perfect, because those are the tissues that have been seen to be affected in CDG, so we were probably on the right track. And in fact, here was kidney, small intestine, and liver. And you remember there was a coagulopathy. The clotting factors were low. In the small intestine, protein-losing enteropathy. And in the liver, there was fibrosis. So again, we thought this was the kind of thing we expected. And so when we actually looked at that, we found, and we were disappointed, and this is the new bad, if you will, that most of the things we assayed for were totally normal. And so after putting in all this time, we thought we had created this wonderful mouse, and the mouse, it just laughs at us. I mean, it doesn't care what's going on. And, we had a slight increase in the amount of calcium, which could be suggestive of some problems with the kidney, and potassium here as well. Liver values that are a little bit high, but again, they're not terrible. And so then we decided in various ways to stress the mouse, and I don't have time to talk about those stress studies right at the moment. But we thought, well, one of the things we better do is to feed the moms mannose. Just in case there are things that we can track, then, of course, by adding mannose to this embryo that now has 15% residual activity, which is considerable, that will be able to correct whatever those deficiencies are that we find later on. Well, here comes the new bad. It turns out that when you put moms on 5% mannose, in fact, what happens is the knock-ins, or as we call them, kikis, the knock-ins are, in fact, being killed in the uterus. So the embryos are dying and being absorbed. And we get no live births from 5% mannose. And what was going on? Well, as you can see here, the wild type, and this is Jonah Nyack that did a lot of this work, wild type series of embryos will look like that, whereas in the knock-in, what you can see is there's a lot of hemorrhaging, and this is in the placenta. The embryos are not very good looking. They're much smaller than they should be, but there's a lot of hemorrhage that's going on, and when we go actually in and do histology on the placenta, we can see it's really bad news, completely deranged, especially for the maternal side. And then what we find is that, in fact, there has been mannose 6-phosphate that has accumulated, not in the embryos, but actually in the placenta. And these numbers that you're seeing here is just with normal maternal mannose. In other words, that's without feeding mannose to mom. So that says that there is a propensity, even when you have normal levels of mannose, but if you don't have enough of this enzyme activity, that you may be at risk for accumulating MAD6-phosphate. And there's a slight decrease under those conditions of normal embryos being born. They said, well, all right, let's lower the concentration. Now what happens? Well, at 0% mannose in the drinking water of moms, there's really not much difference between the knock-ins and the wild type. And if you go to, you drop it down to 2%, you see that there is about 33% that survive to weaning, and the litter size is reduced. And if you go to only 1%, that in fact 61% survive. So this means that there is a dose-dependent effect on the ability of these embryos to survive. So you shouldn't go just drinking a whole bunch of mannose, because this could have deleterious effects. And we said, well, you know, thank God that they were born and they were normal. But then it turns out that some work that Jonah and Bonin have been doing, that's what a normal mouse eye looks like. Turns out that in these guys, they turn ugly. Almost 50% of the mice that were put on 1 or 2% mannose now get these very abnormal eye defects. And this is called, if you look back here, the eyes are shut. That's anophthalmia, cloudy eyes, cataracts that you probably heard about. And there are small cloudy eyes, microphthalmia. And so we did some histology on this, and we find out that, in fact, the eye looks like it's almost being eaten. And we're still working on the kinds of cells that are appearing to attack the eye and absorb it. And we think this is a developmental issue that may occur in about mid-development. We're going back and doing the time course on it now. But as you can see, here's what a normal eye would look like. This is the lens, and the retina is back here. And in fact, now when you look here, you see this accumulation of these dark cells. And you say, oh, it looks like the retina has just expanded. Well, that's not it. We've checked for retinal cell markers, and it's not that. We still don't know what kind of cells they are, because we've done a number of markers. We still don't have that nailed down. But as you can see, on our normal knock-in mice without mannose, this is looking fine. You put them on with mannose, and here is an eye that has been almost totally absorbed. Here is one that is well on its way there. So what does this tell you? Well, there is mannose toxicity. And it is probably, we're still measuring this, it's probably due to mannose 6-phosphate, and we're going back and measuring more in the placenta, and that's due to ATP depletion. So adult mice are not adversely affected by mannose. I mean, they're fine. You can put them on mannose all the time. But in the embryos, it's a different story. So is this something that is species or mouse-specific? We don't know. What about potential problems? Does that have any implications for people? Well, you can imagine that at-risk mothers, in other words, a mother who's had a child with CDG-1b is going to say, well, I'm going to take Mannose for my next baby just in case there may be some problems for my next child. I'm going to drink plenty of Mannose so that there's no problems. And these results say you should put the brakes on. You should not use Mannose indiscriminately. And that's important because there are healthy pregnant women out there who may actually be taking Mannose for dealing with urinary tract infections, because you can buy Mannose at the health food store. And it's a multi-million-dollar industry of people drinking Mannose. And why is that? Well, you just pee it out, and it turns out that the bacteria, E. coli, pathogenic E. coli, will bind onto the epithelial cells through a Mannose-dependent linkage. And if you have enough Mannose coming through, then you simply are washing off the bacteria and preventing their binding. So, one has to be careful in looking at this. So the point, I think, from this is that ongoing basic research is essential, if you will, to find the sweet spot. And the sweet spot is really that place that we can go and know that we're at the right amount of Mannose. And Max, our friend, got lucky. That was a little boy in the slide that I showed in the Lederhosen. He recently had an episode of where he had come into the hospital and was having diarrhea and vomiting, and he wasn't able to hold Mannose down, and he was given it intravenous. When that happened, he went into an energy depletion and had an abnormal EEG and sort of went into a stupor as though he was being energy depleted. Well, he was probably running into the same thing, which is called the honeybee syndrome, that we're seeing in these patients and in these embryos. So one needs to be careful even in safe food. So I'm going to just change gears here for the last couple minutes and talk about the growth of congenital disorders of glycosylation. This is the accumulation of these disorders over the last especially over the last decade, and you can see that there are a large number of them that have come along, and this only goes back to 2010, to last year. And we know that 1 to 2% of the genome encodes proteins that are going to be relevant to glycosylation. Well, it turns out that if we're going to understand a lot of the patients that are now starting to come in, we have to have more interactions here in the U.S., and that can come in several different ways. One is in diagnosis of these patients, and of course a lot of that's awareness. And Emory Genetics is doing a clinically approved test for 25 known CDG genes. So that's good. And we're now starting to get into exome sequencing to try to find mutations in the unknown genes. Mayo Clinic does biochemical diagnosis, and Greenwood Genetics Center does both gene and biochemical. In terms of the basic science, it's still, there are not a lot of labs working on it. We're certainly, of course, doing it here. There are scattered studies, such as in Joe Gleason's lab at UCSD, that have made discoveries in CDG, but there are very few places that are actually focused on CDG research. We now have a zebrafish consortium because the zebrafish looks like they may be a very good model for different types of CDG. And that was in part of the American Recovery and Reconstruction Act. So in terms of patients and potential therapy, at Sanford Health, they're now beginning to try to coordinate networks of rare diseases, and David Pierce is heading up that effort. So our connection with Sanford and Sioux Falls is a real benefit because they have a large medical establishment. They're making a commitment to rare disease, and the head of the CDG family network also lives in Sioux Falls, South Dakota. And in Sanford-Burnham in Florida, we're now beginning to explore exome sequencing, and I'll go on a little bit more about that. So these are the currently known disorders, but given the kinds of things you've heard today, the exploration, the quantum leaps that are being done right now, I think this is what it's going to look like in the next few years. I think we're going to find a boon of discoveries of rare diseases. It's just going to explode. Part of the problem is going to be the other parts you've heard, is that we're going to have to find ways to interpret this. Just having the gene mutation is not enough. You're going to need to be able to go in and characterize this physiologically, biochemically, cell biology, to show that that mutation is in fact what you think it is, and that it's actually causal. And we have about 50 CDG-X patients, and we know that at least in looking at 10 of them, only one of those has been in a known type. So a lot of new diseases are yet to be discovered. So what's our vistas? Well, in the past, I would say 15 years, we've had biochemistry leading us to the genes, and that's worked out pretty well. But that approach, I think, is nearly saturated, not completely, but nearly. And so now genetic mapping is the way to go. But since most of these patients have mutations in don't have mutations in known genes, we're going to have to turn to exome sequencing. And that is certainly going to reveal hypothetical genes, misannotated genes, and genes that you go, well, what do we do with this one? And here's where I think that glycobiology will play a really important role, because even though the informatics is challenging, now we can turn it around and say only 1 or 2% of the genome is probably devoted to glycosylation. So if we spot something in there that we can make some physiological connection to, then we have a better chance of finding that defective gene. So the biochemistry and cell biology is going to be necessary to confirm the pathology, but I think there are great opportunities. And glycosylation disorders, I think, will provide us scientific discoveries, because this is the way it goes. You discover something in the clinic, you bring it back to the lab. You have it in the lab, you take it into the clinic. And then the important part is sharing it with all of the stakeholders that we've got and that you heard from today. And so our acknowledgments go to the people who are currently in the lab: Vandana and Jonah, and Bobby, Mia, Gita Sri Krishna, Miriam, Ping, Maria Stell, Francesca, Emily. Thanks for all your hard work. You guys are doing great. And in Lake Noters, Greg Roth, Leighton Smith, and Thomas Chung here, and Nick Cosford have been very active. And I want to acknowledge our support from the Children's Health Research Center, the Rocket Fund. We would not even have dreamed, let me tell you, we would not even have dreamed of going into these areas of therapy if it hadn't been for the Rocket Fund. I mean, without it, we'd have been square one. And these are our angels on the right. They're our inspiration. So with that, I think I'll close.